Hello, all. We are going to be diving into chapter 3 today. All right, so let me get that pulled up. All right, so for chapter 3, we're going to go over carbon skeletons and functional groups, and then macromolecules, how they're made and broken, and then the different types of each macromolecule. Carbon is a very important atom, an element. Um, it is the main building block of life. Um, our cells are 72% water and 25% carbon compounds. Um, so carbon is important. Carbon is versatile. It can create and make four covalent bonds. Many molecules have nonpolar carbon hydrogen bonds, and this is because they have similar electronegativities. Okay, um, and they're very stable. So here are an example of three ways in which you can assemble organic molecules. You can do the structural formula, which is like the stick with the letters. Then you can do the literal ball and stick model, and then you can do the space filling model, where instead of sticks, the little balls are just directly attached to the center molecule of carbon, in this instance for methane. What do you need to know about carbon skeleton diversity? You need to know that they can vary in length. They can vary in positions of double bonds, the locations of the double bonds. They can have different branching ways, points, or shapes, and they can have different types of rings with different locations of double bonds, for example. Okay, so form really affects function here. An example of this is thalidomide, and this was prescribed to women to reduce morning sickness in the 50s and 60s. But unfortunately, it actually caused severe birth defects, because and so that's not great. And that was the isomer, specifically, was causing the specific birth defects. And an isomer is when something has the same structural or same chemical formula but a different structural formula. Here, where you can see here, we have a different bond here than we have here, and that's what's going to make the difference. So iso means same. Isomers are when they have the same atoms in different arrangements. My favorite isomer is glucose and fructose, where just one of the double bonds is in a different spot. And it's specifically why fructose is more sweet than glucose. Kind of neat. Stereoisomers differ in how the groups are attached to each other. So, like, they're often mirror images of one another. So for stereoisomers, go back here for a moment, usually one of two of them is an active drug, and they're really difficult to separate from each other. For example, there's an ineffective, S-ibuprofen is effective for pain and inflammation, but R-ibuprofen, its model image, is not. And the same is true for R-albuterol and S-albuterol. So, functional groups, these are small chemical changes that result in big biological ones. And their properties are determined by the carbon skeleton that they are attached to and what the functional groups are, the number of them and how many of them. So, here are the functional groups. This is a nice little table that has the functional group in this far column, the structural formula here, an example, and then the where the location of where they are found, most likely found, and then the bond. There's a big P or an NP to indicate polar or non-polar bonding. Okay, carboxyl. So this is an example of one of these groups. Okay, so this usually binds with carboxylic acids. An example here is acetic acid, and it acts as an acid. Okay, aminos. These are going to be found on or is often associated with amino acids. They can act as a base. They can pick hydrogens up from water. So your macromolecules. First, when we build a macromolecule, okay, we're putting two smaller molecules or two monomers together, okay, and this is going to result in what's called a dehydration reaction. So what happens when you're dehydrated? You're lacking or losing water, right? So in dehydration reactions, when we build macromolecules, we're going to actually lose water from that reaction. Here, when we're breaking down macromolecules, we're going to experience hydrolysis or digestion of molecules. And this is where we actually use molecules, specifically water, to break down a bigger molecule, to burst the bonds. And so hydro means water, lysis means to burst. And so when you're breaking down molecules, those molecules undergo hydrolysis, or hydrolysis water is used to burst the bonds connecting those molecules together. All right, carbohydrates, our first macromolecule, incredibly important. Do not ditch carbs. They are your friend and you need them. You need them for energy because your brain consumes 75% of the glucose in your body. And that means you need a lot of glucose, and carbohydrates break down into monomers, which are simple sugars like glucose. So we need those monomers to build these carbohydrates. So you do need your carbs. They're important. Anything in excess, not great, but you do need them. So we have simple and more complex sugars. Simple sugars like glucose and fructose are monosaccharides, meaning they're just one single sugar. Mono means one, saccharide means sugar. Okay, so monosaccharide means one sugar. Disaccharide. Di means two, and saccharide means sugar. So this means two sugars. This is like sucrose, which is table sugar, a combination of glucose and fructose. Woohoo! Then we have our polysaccharides. Poly means many. So this is going to be many small sugars attached together. These are big sugars. And these are things like starch. Okay, I'm going to let you watch this in your own time. Monosaccharide structure. Sugars form rings in aqueous solutions. We talked about dehydration reactions. Polysaccharides. So these are for energy storage. In plants, we use starch. In animals, we use glycogen. You need to know this. They're also used for structure. So cellulose is used in plants, and chitin is used in the cell walls of arthropods and fungi. That's why a mushroom snaps when you break it, or a cockroach crunches. Okay, you need to know all of those energy storage and structure molecules: starch, glycogen, cellulose, chitin. Know them. Here's an example of looking at both cellulose and starch, and you can see how incredibly similar they look. They're not, completely identical, but they're very, very similar in terms of the monomers that they're composed of. Cellulose is the most abundant compound on the planet, and herbivores, those lucky suckers, can digest it, but carnivores and omnivores like us cannot. That's why we eat meat to get our nutrients and proteins, but we also need to eat vegetables for our roughage. Why? Because you have these things on the inside of your intestines called villi. Okay, they're like these little finger-like projections that come off of it. And if those don't get stimulated when food passes through, you're going to end up constipated because your poop is going to get built up because there's nothing stimulating those villi to cause mucus to be secreted to help move your stool along your intestine. So you need to eat cellulose and roughage that we can't digest because it stimulates those villi on the sides of your intestines to go ahead and release that mucus, in which case you won't be as constipated. Lots of polysaccharide diversity. I already mentioned the isomers of glucose. In terms of structure and function. Proteins. Okay. Oops, sorry. Proteins. They act as enzymes. They are important for structure. They're important for carriers and support, receptors and binding, contraction, signaling, and storage. Lots of stuff. Okay. Proteins at their basis are amino acids connected to peptide bonds. Basic things. There are 20 different amino acids. Okay, a monomer is an amino acid in this for a protein, and a polymer is a polypeptide, which is many amino acids. Okay, they have these funky 3D shapes. Okay, amino acids. They have a central carbon, an amino group on one end, a carboxyl group on the other, and an R side chain, which is like another type of functional group. When we build proteins, we're working with hydrolysis or dehydration reactions. We have nonpolar amino acids. Okay, and we have polar and charged amino acids. So there's only 20 amino acids, but the different combinations that we can use are enough for lots and lots of life on Earth. So protein structure depends upon function. So our proteins are twisted and coiled into unique shapes. You can see this in one of these photos here using X-ray crystallography. Primary protein structure is the order of amino acids, and this is determined by their DNA. So this is the amino acids connected to each other by peptide bonds. An example of a change to this order of amino acids, okay, is where if we substituted for glutamate, valine, okay, we end up with sickle cell anemia, where we have changed our instructions of how to make our protein, hence we end up with a different shaped protein product. Secondary structure is going to be either or both of alpha helices or these beta pleated sheets. Okay, there's going to be hydrogen bonding between the backbones, and we haven't involved these R groups yet, okay? So now we've got our amino acid chains, now alpha helices, beta pleated sheets, or both. Then we have what are called hydrophobic interactions that take place in the tertiary structure. This is where those functional R groups come into play, and we have lots of these hydrophobic interactions that are going to start to change the shape of the protein, creating these disulfide bridges between the amino acids. And then your quaternary structure involves more hydrogen bonding and more hydrophobic interactions and lots of bending and shaping to get the protein into whatever shape it needs to be in to do its job. So this is a great slide to study. Okay, primary sequence, amino acid sequence and peptide bonds determined by your DNA. Secondary structure is alpha helices, beta pleated sheets, or both, as a backbone held together with hydrogen bonds. And then tertiary structure is where we have R group interactions and both hydrophobic and other hydrogen bonding interactions occurring to shape us into where we need to be headed. And quaternary structure is where we have multiple polypeptides. We have our both hydrophobic and hydrophilic interactions going on, and we're getting into the exact shape that we need to be in. Now, how do we change the shape of a protein? We denature it. Okay? Why does it get worrying when you get a temperature of 104 or 105? Because your proteins will fall apart. No one wants that to happen. Okay? And they disrupt the hydrogen bonds. So pHs, temperature, and salts can actually have a really big effect on your proteins. Nucleic acids, last but not least, these are our storage and hereditary storage information molecules. We have RNA, ribonucleic acid, which is single-stranded, and then we have DNA, which is deoxyribonucleic acid, which is double-stranded. Okay, the monomers of these nucleic acids are our nucleotides. Nucleotides have a nitrogenous base. We'll get to what those are. A pentose or five-sided sugar, ribose in RNA, and deoxyribose in DNA, and a phosphate group. This little phosphate group off to the side here. Your nitrogenous bases, you have the purines and the pyrimidines. Okay, your purines are adenine and guanine, and your pyrimidines are cytosine, thymine, and uracil. Now, thymine is used in DNA only, and uracil is used in place of thymine in RNA only. So, when we're building these, we have a sugar to phosphate backbone that are connected by these phosphodiester bonds, and the nitrogen bases just kind of hang off the side. Okay, so here's a look at your DNA. So here is your A's, T's, C's, and G's. Okay, so our adenine matches with thymine, and our guanine matches with cytosine. In uracil, in RNA, our adenine matches with uracil. ATP, okay, this is adenine triphosphate. This is the energy molecule for cellular processes. It is composed of a molecule of adenosine and 3 phosphate molecules, hence adenosine triphosphate. Now, when we go to phosphorylate something or give it energy, we are going to take one of those phosphate groups and we're going to shove or give it to whatever we want to have that energy. We are then going to be reduced down to adenosine diphosphate because we'll only have 2 phosphate groups left. Lipids, last but not least, but they're my favorite. These are long hydrocarbon chains of carbons, hydrogens, and oxygens. They contain the fats, the phospholipids, and the steroids. So, fats. Fats are a molecule of glycerol and a fatty acid, 1 to 3 fatty acid tails. Okay? Your fatty acid has a COOH group at the head, okay, or carboxyl group at the head there. Excuse me. Sorry about that. I had to stretch my neck a little bit. So we build our fat molecule. We have 3 fatty acid tails and a molecule of glyceride. This is a triglyceride. Why? 3 fatty acid tails, molecule of glycerol, triglyceride. Fats store energy. Okay, so all of the organs in your body are lined with a thin layer of fat. We all have a thin layer of fat lining underneath our skin to help keep us warm. Okay, so cushions are organs, so they're not like smacking into each other when we're running around. And it helps to keep us warm. Yay. Now, saturated fats and unsaturated fats. I have a thing here. Saturated fats are solid at room temperature and have a single bond. So hence, if they don't have a double bond, they have a single bond. Okay, so saturated fats, solid at room temperature, like butter or lard, and have a single bond. Unsaturated fats are liquid at room temperature and have a double bond, which causes kinks. Again, saturated fats, solid at room temperature, single bond. Unsaturated fats, liquid at room temperature, double bond that causes kinks. Okay, saturated fat, bad, not entirely. You need some saturated fat, but not a lot. And then you need lots of unsaturated fat. Yay, trans fat's bad. And trans fats are bad because they are highly processed, and the process the processing that makes them the way they are causes your bad cholesterol to go up and your good cholesterol to go down. Second type of lipid is the phospholipids. So there is a phosphate head and a fatty acid tail here. Okay, your phosphate head is both negatively charged and hydrophilic, meaning it likes water. And your fatty acid tail is hydrophobic, meaning it doesn't like water. Now, your phospholipids create a barrier in the water and they define the inside versus the outside of your cell. Oh no, I don't know what's happening here. Whoop. Hang on, we are having technical difficulties. Well, we're almost done. See if we can get through them. All right, last but not least, we have the steroids. Okay, let me see if I can get this back into, nope, there we go, the steroids, okay, which are, they have a skeleton of 4 fused carbon rings. They are helpful in the cell membrane, like cholesterol is a big component of the cell membrane. Cholesterol is also a precursor for sex hormones like testosterone and estrogen, which do amazing things for your body. And testosterone is not a male hormone, and estrogen is not a female hormone. They are hormones that all sexes have at differing amounts, with males tending to have higher levels of testosterone and females tending to have higher levels of estrogen and progesterone, but not always. I'm an endocrinologist, so you are always going to hear me say but not always, because there are always exceptions. Too much cholesterol, though, can lead to cardiovascular disease, which is not a great thing. You will watch this little importance of hormones video. Actually, if you don't have this video, you can't.
Dear gentlemen, science has a confession. Your life actually began as a female. Well, phenotypically or physically speaking, that is, though some prefer to call this stage gender neutral. As you might know, when 2 people mate, they each combine half of their genetic information together, 23 chromosomes each. One pair of these chromosomes helps to determine your sex. If you're female, you contain 2 X chromosomes, one from your mother and one from your father, while males have 1 X and 1 Y chromosome. At the moment of conception, you were of course male or female based on your genes, but the Y chromosome doesn't kick in right away. In fact, the first 5 to 6 weeks of embryonic development are attributed to the X chromosome alone, and females grow from embryo to fully developed through the influence of only the X chromosome. So what does the Y chromosome do? Once activated, one of the genes on the Y chromosome, known as the SRY gene, has 2 functions: to inhibit certain features of the X chromosome and to impose through dominance the physiological traits that a male must have. The ovaries descend and become the testes, and the labia fuses to create the scrotum. If you're a male, take a look. Though it's more pronounced in some men than others, you'll likely notice a line or ridge of tissue on the scrotum, which is called the scrotal raphe. The skin of the scrotum and penis are zipped up like a zipper as the layers of cells develop in the embryo. And the male variant of the clitoris? Yep, that's the penis. If the Y chromosome does not become activated for some reason, then the female phenotype or physical appearance will persist in a male. While we're at it, this is also part of the reason males have nipples. The nipples form before the activation of the Y chromosome and SRY gene, and thus remain through development and life. But you don't develop breasts. Sorry. Got a burning question you want answered? Ask it in the comments or on Facebook and Twitter, and subscribe for more weekly science videos.
So our ASAP Science guys are awesome. I love them so much. They make some great videos. Cholesterol, whoopsie daisies, so again, really important with hormones, important component of the cell membrane. It's a fluidity buffer. It keeps them fluid and flexible in the cold, which is really important. And it also keeps lipids from becoming too fluid when it's really hot. All right, great little table here to review all of your macromolecules. And that is going to do it for Chapter 3 for us. So thank you so much for your time and attention. I appreciate it. And I'll see you in the next lecture.